Hi everyone, and welcome back to another episode of Science. In this episode, we're going to cover a little bit of the basics of using a microscope and what is a microscope. In this topic of cells, you'll be using the microscope fairly often actually, having a look at structures and objects that are much smaller on the scale of what your eyes can actually see. So a lot of the time we'll be viewing cells under the microscope. Like with any new topic, I like asking questions. So I wonder, or have you ever wondered, what your skin and hair looks like when it's magnified really closely? Why plants are green, but humans are not? Why do animals need skeletons, but plants don't need skeletons? These are just a very small number of some of the questions you'll be able to answer by the end of this topic. These three cool images that I've got here, these were all taken with a very strong microscope, an electron microscope. So this guy here, I can't quite remember what he was, maybe some kind of mite or something like that, but you can see his jaws munching away on possibly dead skin cells. You've got sugar and salt granules here, and you've got what's here, I believe is a tardigrade. And these guys live all over the place, in ponds, in your backyard, everywhere. They're very, very hardy creatures. Let's take a step forward from here. Okay, so in this topic, we'll explore using microscopes. That's going to be a major area of this topic. We'll also learn about plant cells and animal cells. We'll learn how cells form the building blocks of life, and we'll have a look at specialized cells as well. Your body is full of specialized cells. Your skin has specialized cells, your eyeballs have specialized cells, your heart, your brain, all these specialized cells that perform specialized functions. On the screen at the three images I have here, all of you used to look like this. So this is an egg cell and this is a sperm cell. And once the sperm fertilizes the egg, a zygote is born that we'll call a fertilized egg a zygote. And so all of you used to look like this image. This would be the earliest image taken of you. You are a single cell within your mother's womb, and from one single cell you double into 2, double into 4, double into 8, so on and so forth until you are born as a baby. And when you're born as a baby, you continue to grow into adulthood. So we'll have a look at how this happens in more detail throughout this topic as well. But I always love reflecting on this image in particular because how cool is it that you're looking back on the earliest form of yourself. All of you would have just looked like this singular cell. And now you've grown up enough to learn and look at what you used to look like as a single cell. I mean, to me, that's weird. And then eventually you multiply into a number of cells, and those number of cells start to specialize in making specific tissues within your body. We'll have a look at that in later episodes as well. Here's some more microscope images. So this one is it's a virus. I think this one is actually a bacteriophage, where this particular virus actually infects bacteria rather than people or animals. This is an image of a fly, and these are real images taken with an electron microscope. This is Velcro. This one, I believe, is a moth, and you can see the moth has, or it could be a butterfly, I'm not too sure. I think it's a moth. And you can see his tongue sort of rolled up, and he stretches it out to drink nectar from plants. And I think these guys might be lice. So you can see there the claws that are hanging onto the individual strands of hair. Pretty nasty looking creatures when viewed up close. And the internet has heaps and heaps of really cool looking images of electron microscopes. So if you're curious to see more stuff out there, definitely hit the Googles and search up some cool electron microscopy. Okay, so this is the microscope you're going to be using in class. There's a few structures that you'll need to be familiar with in order to use it. First off is the eyepiece. So this is where you look into. And you've got a bunch of lenses that flow through this arm structure here. And at the bottom of it, you've got what's called an objective lens. Our microscopes generally have 3 objective lenses. We've got the arm piece here. So generally when you hold the microscope, you can hold it from the arm as well as another hand underneath the base. We've got some stage controls. We've got some focusing knobs. We've got a coarse focus and a fine focus here as well. We've got a light that shines a beam of light up through the specimen and then up into the objective lens, and then that light travels up here, up the eyepiece into your eyeball. And the different size objective lenses represent different magnifications, and I'm going to go through that as well. So your specimen sits on this stage here, and you can actually move the specimen about using the stage controls. But this is a general overview of the structure of a microscope. And I've got a side-on picture here as well. Here's my eyepiece, my arm, my adjusting knobs, my objective lenses, the stage where you put your specimen, the diaphragm is how you control how much light you have, and then a light source, and then a base. So once you start using this thing, it's a pretty cool tool to use. Some of you may even end up buying one for yourselves to look at stuff at home. Okay, so magnification, let's talk a little bit about this. So the ocular magnification, which is the eyepiece. So if I come back here, this eyepiece here has a lens. And that lens always has a ×10 magnification. Okay, so on the microscopes you're using, the ocular lens will always have a ×10. And then we've got 3 objective lenses. So if I go back, we've got 1, 2, 3 objective lenses. I know this one's got 4, but the ones you'll be using will have 3 objective lenses. And each of the objective lenses has a different magnification power. So if I come here, the weakest one is ×4. So if you combine your ocular magnification with your objective lens magnification, you end up looking at your specimen at ×40 magnification. So you're seeing it enlarged by 40 times compared to your own eyeball. Then we have a ×10 magnification, and if you combine that with your ocular lens, you're looking at your specimen at ×100 its size. And then our strongest one and the trickiest one to focus with is the ×40 objective lens. And that's this big guy here, the big blue one here. That's a ×40 magnification. And so here you're looking at specimens ×400 magnification. Really, really up close. And it's some of the detail you can see, if you can get the focus right, is just awesome. I can't wait to show you. So what can you see with microscopes? So here's a scale which you can look at. So with the unaided eye, you can go down to about a hair width, but anything thinner or smaller than the width of a hair width is a bit tricky for our eyes to see, and certainly we can't make out any details. When you start to go below the width of a hair width, you need a light microscope, which is what you're using, light microscope. That's what you're going to be using in this topic. And with a light microscope, you can see most cells, so plant and animal cells, and maybe you can see some large bacteria, but this will be very hard for us. Mostly we're going to be having a look at plant and animal cells, and we'll be looking at hair fibers, or hairs and clothing fibers underneath the microscope. You can put bugs and stuff under the microscope and ants. That's the kind of scale we're going to be working with. If we're talking about an electron microscope though, an electron microscope can go much, much smaller, all the way down to the nanometers, right? So we're nearly looking at atoms. Electron microscopes are very powerful indeed. And that basically wraps up this introductory lesson. From here, I actually want you to work on the Microscope Goodness booklet that I would have handed to you or will hand to you. And after you've finished this off, come get it signed by me, and then you can jump onto Education Perfect and have a go at the microscope modules there. Thank you for tuning in, and I will see you on the next episode.